everyone, welcome back to Molecular Biology and Biotechnology with Lucy. And today we have a very special guest and friend called Vanessa, and she's going to be taking us in our next lesson. So without much ado, karibu.
Asante Lucy, asante sana. Thank you very much. I am Irumva Vanessa, as she said, and I'll be taking you through protein structure. It's a very, very good pleasure to be here with you, and I really hope that we have fun in this amazing and exciting world of molecular biology. And also thank you, Lucy, for inviting me and giving me this opportunity.
You're welcome.
Hello everyone, and welcome again to Molecular Biology and Biotechnology with Lucy. As Lucy has introduced me, I am Irumva Vanessa, and I am a molecular biologist. Today, I will take you through the protein structure. I really hope that we will have fun in this exciting and amazing world of biology, and especially molecular biology. So let's go. As some of you may know, simple proteins are made up of peptide bonds, and those bonds in a particular protein are folded in the same way. This is what we call chain conformation. The unique chain conformation of a given protein is influenced by many weak forces that can be discovered by these ionic bonds, hydrogen bonds, etc. Conjugated proteins can be found, which are proteins that contain other non-protein groups that are called prosthetic groups. Proteins have different levels in their structures. The first one is the primary structure, followed by the secondary structure, then the tertiary structure, and finally the quaternary structure. Now, let's go a little bit deeper to see which one of these structures. First of all, the primary structure is the sequence of the amino acids that are linked together by the peptide bonds. Then come the secondary structure that can be the alpha helix structure or maybe the beta pleated sheet. The tertiary structure is also another level of the protein structure and is considered as the three-dimensional shape of a protein. Then we have the quaternary structure, which is found in oligomeric protein or proteins composed of more than one subunit. Like here we have the first subunit, the second subunit, the third subunit, and the fourth subunit. Each of these subunits will come together with others to form the quaternary structure. Now, the primary structure. The primary structure is the amino acid sequence, as we have said. As we know, peptides have less than 30 amino acids, while proteins are the sequence of amino acids in number of 200, between 200 to 1000, joined together by amide bonds, also called peptide bonds. Here, this beautiful picture, we can see different amino acids in different color. Here it's an amino acid, this one, also this one, and in between they are linked, but what we have called the peptide bond in black, please. Here in black, these are links between these amino acids in these different colors. Then the secondary structure, it is the structure that is found with regular recurring arrangements in space of adjacent amino acid residues in a polypeptide chain. Most of the types of the secondary structure are the alpha helix and beta pleated sheet. Now, the alpha helix is a right-handed helix, and the helixes are joined together by intrachain bonds between the carbonyl oxygen of an amino acid residue and the NH hydrogen of the fourth residue down the chain. Let's see with a picture. Here we can see this is an helix that is found in the alpha helix of a protein. Now the secondary structure that is in form of beta-plated sheet. It is arranged side by side, resembling a piece of paper like this one, folded into plates. Like a helix, the beta sheet uses the full hydrogen bonding capacity of the polypeptide backbone. However, the hydrogen binding occurs between neighboring peptide chains rather than within one. Here we can see the secondary structure that is in the beta-plated sheet. Here we can see it's like a piece of paper that we have plated. Great. Now the tertiary structure. It refers to the complete 3D structure of an entire polypeptide, and it usually involves the packing of structure elements. Being here, you can ask yourself, why do we need to know these structures? Why is this really important? Please stay tuned to know why we need to know the structures of proteins. In the next lesson, we will see that there are diseases that are linked to the fact that proteins have not attained their tertiary structure or are not in their real or in their effective tertiary structure. Stay tuned, please. This is an example of tertiary structure of the sperm whale microorganism. We can see here it is a protein in its tertiary structure in its three-dimensional shape. Here also it's another protein called cytochrome C. As we know, the cytochrome C is a heme protein that is found in the mitochondria intermembrane. Another example is the lysozyme, which is an enzyme. Then let's see the quaternary structure. As we have seen, the quaternary structure is found in oligomeric proteins that are composed of more than one subunit. Then the quaternary structure refers to special arrangement of subunits. Here, within the proteins, subunits generally associate through non-covalent. These are links between those different subunits, interactions, and in some cases disfied bonds. Here the quaternary structure of different proteins. These beautiful pictures are showing us different quaternary structure of protein. Here we have the E. coli fumarate. We can see we have many different subunits in different colors. This one, the first one, the second one, the third one, the fourth one. Here we have 4 subunits and here we have 3 subunits. Here also we are also seeing the quaternary structure of these different proteins, while in a human or maybe in a bacteria. These different proteins can be bound using different bonds. We have covalent bonds, coordinate covalent bonds, ionic forces, hydrogen bonds, van der Waals forces, hydrophobic interactions, and also electrostatic charges. Thank you very much for being in this lecture, and please stay tuned and subscribe to this YouTube channel to know more about molecular biology. In the next lesson, Lucy will take us through folding, unfolding, and misfolding of proteins. Here we will see what is the consequences that can happen when proteins are not into their right structure. So thank you and stay tuned.